Hello friends, welcome to my channel Med Simply Learn. Today's video we will be discussing about enzymes, their nomenclature and classification. So let's start the video. So first of all, a brief introduction about enzymes. Enzymes are important biological catalysts. They accelerate the reaction rates in living organisms. Coming to nomenclature, let's have a brief historical overview first. So earlier, enzymes were named arbitrarily or randomly based on the substrate or some functional terms, and there was no systematic approach. And these names were known as trivial names or the common names. Examples include pepsin, which is derived from Greek word pepsis, which means digestion, as pepsin has a role in digestion. So based on the function, it was named such a way. Trypsin, another important digestive enzyme, name was derived from Greek word tryptin, that is to wear down. So these were the trivial names, but these names were simple and practical but had various limitations. So let's discuss the limitations of trivial names or the common names of the enzymes. First limitation was lack of specificity. So the trivial names were very non-specific, did not tell about the enzyme function. Secondly, inconsistencies in naming. Some enzymes have multiple trivial names. Example like lactase, also called as beta-galactosidase, and this was led to confusion among the researchers. Another important limitation was no indication of reaction mechanism. These names did not have any indication about what type of reaction that particular enzyme is catalyzing. Then misleading names. Some of the names were misleading. For example, invertase. It sounds like it inverts something, but actually it hydrolyzes sucrose into glucose and fructose. And finally, there was no systematic classification. That is, there was no uniform method of classification, which was making it difficult to compare enzymes systematically or the worldwide. There was no uniformity. So to overcome all these limitations, International Union of Biochemistry and Molecular Biology Enzyme Commission was established in the year 1961, which gave the IUBMB nomenclature for the enzymes. Now, the goal of this commission was to create a comprehensive means detailed, unambiguous, that is without any confusion or error free, and standardized nomenclature. So this International Union of Biochemistry and Molecular Biology was constituted in 1961 to form a comprehensive, unambiguous, and a standardized nomenclature system. Therefore, they devised the systematic nomenclature of the enzymes, which is the modern day nomenclature, and classified the enzymes into various classes and subclasses, which we will be discussing. Now, according to this nomenclature, enzymes have systematic names, and systematic names consist of two parts. The first part contains the name of the substrate, that is, the enzyme is acting on which substrate. That is the first part of the name. And the second part of the name ending in ASE suffix indicates the nature of reaction, what type of reaction that particular enzyme is catalyzing. Let us try to understand this through this example. The enzyme name here is alcohol dehydrogenase. So the first part, that is alcohol, is the substrate on which this particular enzyme is acting. And the second part, dehydrogenase, tells us about the nature of reaction, that is oxidation of from alcohols. So this is about IUPAC nomenclature. Now this IUPAC nomenclature devised a specific number system for the enzymes. Each enzyme was given a specific 4-part numerical value. It is represented as EC, enzyme class X dot X dot X dot X dot. Each of these digits, which are 4, we can see here, have a specific meaning. Let's try to understand. The first X denotes the main class. There are 7 classes as per this classification, so the first digit is telling about the main class of the enzyme, and this class defines the type of reaction that particular enzyme is catalyzing. The second digit tells us about the subclass, and subclass mainly based upon the functional group of on which that particular enzyme is acting. And the third digit specifies the subclass. This third digit will further detail the reaction. They will identify bonds or the groups involved. And finally, the fourth digit is the serial number as per the discovery of that particular enzyme, and it provides a unique identifier for each enzyme. Thus, it gives us the precise identification of that particular enzyme. Now, it may be sound very confusing to all of you when I am trying to explain this, but when we will see through an example, it will make it more clear to you. Now, let's see the example alcohol dehydrogenase in the enzyme, and the EC number is 1.1.1.1. Now, let's try to understand this. Now, let's see the breakdown of this EC number. The first one means it belongs to the enzyme category 1 or the enzyme class 1, which is the oxidoreductases. So this alcohol dehydrogenase, this one means belongs to oxidoreductase class of enzyme, which involved in oxidation reduction reactions. The second one indicates the subclass, and 1.1 subclass is the class in which the enzyme is acting on the alcoholic group. So it tells us about that this enzyme is acting on the hydroxyl group or the alcoholic group. The third one tells us that it uses NAD plus or NADP plus as an acceptor. So it is giving us more details about the reaction. The first told about what type of reaction it is. The second digit told us about the group or the functional group. Third digit further added to the information provided by the first 2 digits about the reaction as well as the group. Here it is telling us about that NAD plus or NADP plus is acting as an acceptor. So, and the final fourth digit is the specific number completes the specific identifier number. So it is 1 because it is the first enzyme identified in the category of oxidoreductases. So this is how the number system is to be understood or interpreted. Okay. So let's discuss the various classes of enzymes based on this classification. The first class is of oxidoreductases. Oxidoreductases are the enzymes that catalyze oxidation reduction reactions, also known as redox reactions. In these reactions, one of the molecule will donate the electrons and undergo oxidation, and another molecule will accept the electrons and undergo reduction. Let's try to understand this. See, here we have A molecule, which is a reduced molecule, that is, it will donate the electrons, while B molecule is oxidized, it will accept the electrons. Now this A molecule will lose electrons and get oxidized, while B molecule will gain the electrons and get reduced. This is how the oxidation reduction reaction occurs, and oxidoreductases catalyze such type of a reaction. Let's see the example. Lactate dehydrogenase catalyzes the oxidation of lactate to pyruvate. Here lactate is oxidized to pyruvate, while NAD plus is reduced to NADH and H plus. And these enzymes play a very important role in cellular respiration, metabolism, and detoxification. And coming to the various subclasses are based on the functional group on which that particular enzyme is acting. For example, the first subclass enzymes are acting on the alcoholic group, example being alcoholic dehydrogenase. Second subclass, the enzymes are acting on aldehyde or the keto group, example is aldehyde dehydrogenase. Similarly, we have various other subclasses depending upon the functional group on which that particular enzyme is acting. Now coming to the second class of the enzymes, which is transferases. As the name itself suggests, transferases are the enzymes which transfer a functional group from one molecule to the another molecule. So the general reaction, we can see one of the molecule, which is the donor molecule, will donate a functional group, and the another, which is the acceptor molecule, will accept that particular functional group. So in this way, that functional group is transferred from one molecule to the another molecule. Example being hexokinase. We all know hexokinase is a very important enzyme of the glycolytic pathway. It catalyzes the first step of the glycolytic pathway, which is glucose gets converted to glucose 6-phosphate. Here glucose is accepting a phosphate group, while ATP is donating a phosphate group. So this way hexokinase, being a transferase, catalyzes the transfer of phosphate group from ATP to the glucose molecule. Now these enzymes have a very important role in cell signaling, metabolism, and biosynthetic pathways. Coming to the subclasses, transferases again are divided into various subclasses depending upon the group which that particular enzyme is transferring. For example, the first subclass is in which the one carbon groups are transferred. Example is methyl transferase, which causes addition of methyl groups in the DNA, leading to methylation of DNA. The second subclass transfers aldehyde or the ketone group. Example is transketolase. It transfers the ketone groups in the PPP pathway. And similarly, we have various other subclasses. The third class of the enzyme is of hydrolases. Hydrolases are the enzymes that catalyze hydrolysis of the chemical bond by addition of water molecule. So the reaction here would be substrate will be broken down into product 1 and product 2 by the addition of water. Example is lactase. Lactase enzyme hydrolyzes lactase to glucose plus galactose. And importance of these enzymes is they have important role in digestion, metabolism and biomolecule degradation. And what are the various subclasses? Here the subclassification is based on the bonds which are hydrolyzed by a particular hydrolase. For example, the first subclass is esterases. So the ester bonds are broken down. Example include lipase. The second subclass is of glycosidases, which break the glycosidic bond, which are the bonds present in the sugars. So example here is amylase. Similarly, we have other subclasses of the hydrolases. The fourth class of the enzyme is that of lyases. Lyases are also causing the breaking of the chemical bonds just like the hydrolases. But here there is no need of water molecule. That is, there is no addition of water. Also, there is no oxidation reduction reaction involved. Instead, these often form either the double bonds or add groups to the molecules. So the reaction would be A molecule is broken down into B and C without any addition of a water molecule. Example being pyruvate decarboxylase, which breaks pyruvate into acetaldehyde and carbon dioxide. And these enzymes have a role in energy metabolism, neurotransmitter synthesis, and hormone signaling. And the various subclasses are based upon the bond which is lysed by that particular lyase. For example, the first subclass will break the carbon-carbon bond. Example, we have just seen pyruvate decarboxylase. Second type of subclass breaks the carbon-oxygen bond. For example, aconitase, which converts citrate to isocitrate in the Krebs cycle. So similarly, we have other subclasses of lyases. Coming to the fifth class, that is isomerases. So isomerases are the enzymes that catalyze the rearrangement of atoms within a molecule, converting it into its isomers. We all know isomers are the same molecules but different structure. They have same chemical formula but structurally they are different. So these reactions do not add or remove any atoms but simply rearrange the structure of the molecule. A is converted to B simply. There is nothing addition, nothing subtraction. So A and B are isomers of the each other, meaning they have the same molecular formula but structurally they are different. And these enzymes have a role in energy production, amino acid metabolism, protein folding, that is they prevent misfolding of the proteins, in DNA replication and biosynthetic pathways. And the subclasses, again we have the first subclass of racemases and epimerases, which interconvert the stereoisomers. Example being alanine racemase. Similarly, we have cis trans isomerases which is the second subclass of this particular category, and we have various other subclasses. Coming to the sixth class of the classification, that is of ligases. Ligases are the enzymes that catalyzes the joining of two molecules. We have seen various classes in which there is breaking of the bonds, but here there is ligation. Ligation means to join. So ligases are the enzymes which join the two molecules by forming a new chemical bond, and usually these enzymes, they need ATP or some another energy source for making such a new bond. So we can see the reaction A plus B gets converted into AB in the presence of ATP, which is providing the energy. Example is DNA ligase, which repairs the broken DNA strands in the DNA replication pathway by forming the phosphodiester bonds. These enzymes play a crucial role in DNA repair, DNA replication, metabolism, and biosynthesis. And various subclasses include the type of bond which is formed by a particular ligase. For example, the first subclass, the bond formed is a carbon-oxygen bond, example being pyruvate carboxylase that converts pyruvate to oxaloacetate in gluconeogenic pathway. Second subclass includes enzymes which are forming the carbon-sulfur bond. Example being acetyl coenzymase synthetase, which activates acetate for the metabolism. And similarly, we have various other subclasses of ligases. So the final class of this classification is translocases. As the name itself suggests, translocase, these are the enzymes that translocate or move the molecules from one place to another across the membranes or within the cells. So substrate inside the cell will go outside through the translocase, or a molecule will reach a different location within a cell. If it is across the membrane, we will depict it like this. If it is within the cell, we can depict it in this manner. Example includes ATP synthase enzyme that moves the protons across the mitochondrial membrane to generate the ATP. Okay, this is a very important enzyme in the energy production pathway. These enzymes are important for, as the name itself, translocase, so they're important for transport of the various molecules within the cell or across the membrane, energy conversion or energy production, and metabolism. And the subclasses include the translocation of molecule based on the which molecules they are translocating. For example, the first subclass, there is translocation of the H plus ions. Example we have just seen, ATP synthase. It produces a proton gradient to produce the ATP. Okay. The second subclass catalyzes translocation of inorganic cations like sodium, potassium, calcium, etc. Example being sodium potassium ATPase. It maintains sodium and potassium balance in the cells. So similarly, we have various other subclasses of translocase enzymes. After seeing the various classes of the enzymes, finally let's see the limitations of this classification. So first important limitation is this system does not give information about the enzyme structure. Okay. Second important limitation is new enzymes are constantly being discovered, so this classification has to be updated on a regular basis to remain significant. And the last important limitation or challenge is some enzymes have varied activities, so how to classify those enzymes? This complicates the classification as we cannot put a same enzyme in two different classes. So this was all about enzymes, their nomenclature and classification. Thanks for watching. I hope you liked the video. Please like, share and subscribe my channel, and in the comment section please do let me know what other topics you want me to discuss. That motivates us and helps to create more informative content for our subscribers. Thanks.